that we have discussed in the interactive session and are good files to refer. Well, now, my plan moving forward is, because the big foundation uh, 2 block is over, is I'm going to do some announcements. I'm going to do one whiteboard session per week, and coming week it is at 5:15 on Tuesday, because I think Thursday 12 was not working twice. So I'm coming Tuesday 5:15 for whiteboard for transcription and protein translation. Please, whiteboards are extra help, right? Everything I address it in the class, so there should be no fear of missing out. Then secondly, students are asking about supplemental session. Now, supplemental session also moving forward, unless there are too many lectures in the week, I will do a quick recap between 10 to 11 just to tell you, okay, what you learned. And then from 11, I will open it for question and answer. So moving forward, the supplemental session will only be for an hour. So let's see now today. Remember that your exam is not Monday, but believe me, how you succeed in any course, especially in biochemistry and genetics, because biochemistry, whether genetics, they are very volatile. You know it today, you don't know it tomorrow. It's best to finish these lectures this weekend, so you're ready for another load coming week. This is not my exam review. I'm just going to refresh you what we learned this week. Next week, I'm going to do exam review and also open questions if students have. So let's see what we have learned. Learned this week. We did 3 lectures, 3 sessions and 4 lectures. We did RNA type synthesis and processing. We did protein translation and genetic code. My pointers are also in line with learning objectives if you can benefit with them. As I told you earlier, any of the help files, whether Zoom supplemental, recaps, they will be available in the MSK1 exam help folder. Please look for it. Your class reps will soon create that folder in the drive, right? So we are more productive. So these pointers are same. For example, as I told you, not just know, can you see my slide, everyone? Yes, can you see my RNA types and synthesis recap slide? Just as I told you, bring in rifampicin, bring in Hutchinson's disease, SLE. You need to know the base, which is the cake, but ice them with clinical application and drugs. Please see that you have only 15 questions for these 4 lectures. Every lecture would be tested, but see that 5 other concepts will be tested more, right? So let's start what we did for the RNA type synthesis and transcription. So see, first of all, I tell you, bring in the ingredients. We did transcription, but transcription requires a different enzyme, which is not same as replication. So what does transcription require? RNA polymerase. Is it a DNA-dependent RNA polymerase? Absolutely. Then what products came out of this RNA polymerase? Ribosomal messenger and transfer RNA. Are these your major RNAs? But in eukaryotes, you have different RNA polymerase to synthesize them. So who synthesizes mRNA? Anyone? Who synthesizes mRNA? RNA polymerase 2. So I told you to write in the same order, RMT. So you know it's 1, 2, and 3 polymerase respectively that synthesize it. First of all, you should know the ingredients. You can't mess transcription with translation. Why? Because transcription also has initiation, elongation, and termination. I'm not teaching you here. I'm bringing key points. So pay attention on them. Apart from major classes, keep an eye on snRNA, microRNA, RNase-P, where you have seen them. snRNA and snRNP were for splicing. MicroRNA we have not done yet, but they have in detail, but they interfere with the function of RNA, and naturally there will be no transcription, no translation. RNase-P was a little bit involved in post-transcriptional processing of transfer RNA. That's why I encourage students to write like this so they don't mess up with the polymerase that synthesize them. Only this subunit 5S is synthesized by RNA polymerase 3. Otherwise, ribosomal RNA is by 1, except 5S is by 3. Please understand that you need to bring in pro and eukaryotic differences for all, whether transcription, whether the subunits, and you know that small versus large. Which one has catalytic activity, small or large subunit? Which one has catalytic activity, which is peptidyl transferase, small or large? Large. Then comes your drugs. Why you need to understand that? Because in translation, you will have drugs that affect the large subunit. Example, anyone? Drugs that affect the large subunit? Bigger classification. Are there drugs that affect large subunit? There is lincomycin, there is macrolide, and there is chloramphenicol. Why do you need to understand that? To connect it with normal. Since large subunit has peptidyl transferase activity, whether it's lincomycin, whether it's macrolides or chloramphenicol, what activity they will affect instead of scanning and misreading? What will be their outcome? Peptidyl transferase activity. So do you understand where I am connecting you? Everybody? Yes? You have to know normal to understand abnormal. Does that make sense? So we distinguish the different type of RNAs. We distinguish pro versus eukaryotes. Now, it's not that you are drawing this structure, but you see a clover leaf structure. This is anticodon arm, this is acceptor arm. You see very unusual amino acids here like pseudouridine. We are talking of transfer RNA. So basically you're recognizing the products of transcription, which is mRNA, transfer RNA, and ribosomal RNA. This is not ribosomal RNA. And please know that this is the acceptor arm which accept amino acid, and we will do some post-transcriptional processing which are template independent. Adding CCA to 3' is also template independent. So first we do what? What do we require for transcription? We do the, remember when you talk about transcription, bases are still AUGC. But what we are doing in transcription, we are synthesizing ribosomal messenger transfer RNA, enzyme needed, RNA polymerase. Are they different enzyme for different RNA? Absolutely. Then should you know their structure? Yes. Unusual bases like pseudouridine you see in transfer RNA, CCA sequence, transfer RNA, anticodon arm, transfer RNA, acceptor arm, transfer RNA. We are not talking ribosomal here. Messenger is absolutely different. If you have any questions, do let me know. Why tell when you were there, understand why do you have antibiotic option? No, there is no clinical benefit of giving one over the other. It is what you need for as an outcome. That's why you need different antibiotics, and also it is dependent on the organism sensitivity. So you need to test antibiotic with that too. It's not that you're giving one over the other, whatever is required. Do you want to achieve 30S inhibition or 50S inhibition? I think we are going into farm here.
Yes, I think so as well.
Yes, right. It's here I'm just trying to bring your attention to that you know these ribosomal subunits, pro versus eukaryotes. What you need to connect right now is which antibiotics affect which subunits and what is the outcome. Does that make sense? Yes, you will be taught about these drugs in detail later in Form 2. When you talk about mRNA, it doesn't look anything like transfer RNA, linear, cap on 5' tail on 3'. Now, please see why am I stressing on that. Later on regulation of gene expression, you will understand how stability is so important for when you add a poly A tail. Stability is more on 3' side, poly A tail, though both are for protection of mRNA. 5' cap protects, 3' poly A tail protects. There's a coding sequence. These are transcribed 5' UTR 3', but they are not translated. Where do they come from? Where are they transcribed from? Exon. So soon I will also introduce what is the central dogma, which is DNA, mRNA, and peptide. But you understand this is not, there's a purpose to add a cap. There's a purpose to add a tail to protect mRNA, because transcription is happening where? Where is transcription happening? In the nucleus or in the cytosol? Transcription is happening in the nucleus. Soon after transcription, you need to transport all these RNAs in the cytosol for translation. For the most protection that RNA needs is the mRNA, because it's linear and can be degraded by exonucleases, both on 5' 3' side. Please bring your prokaryotic and eukaryotic differences everywhere. Pay attention on these major RNAs. You should know, look at their structure, how they are different from each other. But microRNAs, snRNAs, RNase-P, please pay attention. MI and small interfering, please, we have not done in detail yet, but as the name says, interfering means they interfere with the function of mRNA. Please understand the sense strand and template strand and the polarity of mRNA. Please understand that RNA polymerase always moves. So there are only some activities which are in 3' 5' direction, which is this one. RNA polymerase always moves from 3' to 5' direction, and that becomes the template strand. Sense is always 5' 3', template is always 3' 5', and then polymerization of RNA by RNA polymerase is always 5' to 3'. Please understand that this is not just RNA polymerase. So you first of all, you understand it should be 2 when it's mRNA, right? It should be RNA polymerase 2 when it's mRNA. 1 ribosomal, 2 messenger, and 3 transfer RNA. But not every RNA is undergoing post-transcriptional processing. So transcription is not complete until post-transcriptional modification have been made, which is extensive for mRNA, where the cap, tail, splicing introns, including exon, right? So this is now. Students are confused with or are worried about application. No, you don't need to. If they give everything points to, in the end, you have to find mRNA. You know why? Because mRNA has the codons that you need to go to the genetic code table to decode them. So everywhere in the end, you have to find mRNA. In any case, whether it's mutation or you want to see change in amino acid or you want to see and if it's not change. So that is why if they give sense strand, quickly find mRNA, because mRNA is same as sense strand. They may give you template strand. How do you find mRNA? mRNA is complementary to template strand. Polarity, you have to pay attention. First, find out same and complementary. Then pay attention in polarity, because mRNA is always 5' to 3'. So read from 5' to 3'. Does that make sense? But then what about if they give you anticodon? If they give you anticodon, mRNA is complementary and antiparallel to anticodon. So you could be given sense strand, which is on DNA, which you could be given template strand, which is on DNA. Or you could be given anticodon, which is in transfer RNA. But what is your final goal? Is to find codons on mRNA, because that is the only place you can go to the genetic code table and decode them. So understand your final outcome is finding the mRNA codon so that you can decode the amino acid. Does that make sense? Initiation, elongation, termination is also for both transcription, translation. Initiation is always highly controlled. I told you to write them in the same manner, and remember 5S is the only one which is synthesized by 3. There is also mushroom sensitivity that also distinguish RNA polymerase. That is also one way they distinguish different RNA polymerase, by mushroom sensitivity. Sensitivity to of different, but understand that not every RNA is undergoing post-transcriptional processing. So did we discuss all of them for mRNA? Yes. But which one is only which one are template independent post-transcriptional processing? So you have to be very specific. That's all. Genetics is this is a very good grade grabbing exam, please. 2 plus 2 is 4. It's not 5. Secondly, if you go stepwise, you cannot make a mistake. Please understand that the post-transcription processing is also a part of transcription completion. You just don't stop transcription after using RNA polymerase 1, 2, and 3. But Not every RNA is undergoing extensive post-transcriptional processing. We discussed it only for mRNA. So which one is template independent? Anyone for mRNA? What was template independent post-transcriptional processing for mRNA? Only poly A cap? Okay, poly A tail and cap. Yes, correct. Poly A tail and cap are template independent. Splicing is template dependent, right? Then you need transporters always. Trex is unique to mRNA. Please understand that is what I'm telling you is transcription. If you want to complete transcription, know your, yes, no worries. I got it. I got it, Diego. Please understand that transcription doesn't just finish with using different RNA polymerase for different products, but also completing it with post-transcriptional processing and transporters. And then transcription is complete. And then also you don't stop here. You ice it with your icing is your disease and drugs. So have we done rifampicin? What is it inhibiting? What is its specific site of inhibition, rifampicin? Anyone? Is it inhibiting any enzyme and stopping the cell proliferation? No transcription, no translation, cell will not be proliferating. No, rifampicin is RNA inhibitor of RNA polymerase. Yes, bacteria are RNA polymerase. Rifampicin is different. Your 30S is your aminoglycosides, your doxycycline, they are 30S. 30S is different, right? So see, just keep on doing them so you don't confuse them. Does that make sense? So you need transporters. It is initiation, elongation, and termination. Who guides them? Tata box. Tata box guides all RNA polymerase, which is known as Hugnus in eukaryotes, Pribno in prokaryotes. Please know that RNA polymerase moves only in 3' to 5' direction. That becomes the template strand. So which is the template strand here? Top or bottom? Which is the template strand here? Top or bottom? In this question, it is template strand. Where is RNA polymerase? RNA polymerase moves only in 3' to 5' direction. So which is the template strand? Top. It's not bottom, top. It's the top because top is only 3', 5' direction. And then whatever is your choice. Yes, anyone has question? Top is the template strand. Top is the template strand because RNA polymerase moves only in 3' to 5' direction, right? So if you want to find out sequence, I would encourage you to do from sense strand because it is easy. mRNA is same as sense strand and mRNA is complementary to template strand. Yes? Anybody has confusion? What was the oh my God for? Everybody is good? Any confusion? So you can use both the strand to find mRNA. Your final aim is to find out the sequence of mRNA. I would encourage you to use sense strand because sense is same as mRNA is same as sense strand except you have 5'. First find out the sequence, then read the polarity. So that is why your answer should be E, right? Please understand the direction. Anyone has any confusion why people are saying bottom? Don't let the arrow mislead you. Arrow is just showing direction. But polymerase moves only from 3' to 5'. Everybody is good here? Yes? Top is the template strand. Then transcription factors helps. Yes, each one of have their function. Know them. Is capping and tail template independent? Yes. Introns splicing is template dependent. Splicing is template dependent. Some are template dependent, some are template independent. There is a signal. Thus, adding poly a tail, where do you add tail? 5' or 3'? You add tail on 3' end. And there is a signal with, and who does that? It is not RNA polymerase 1, 2, and 3 that add the tail. It is poly-A polymerase that adds the tail. And what signal that poly-A polymerase recognizes? This AAUA and keeps adding the tail. Tail is a number of AAA residues, nucleotides, sorry. Where is tail? 5 prime or 3 prime? And why are you adding the tail? So it's not just memorization. Poly-A polymerase adds the tail. But where? 3 prime side. Why you want to add the tail? You want to add the tail for what? Stability of mRNA. Protection, absolutely. 5 prime is you wear a cap for protection in the sun, right? So cap on 5 prime, we don't wear a tail though for protection, but yes, cap on 5 prime side and tail on 3 prime side. Yes, for tail especially is for stability. Then bring in your actinomycin D, rifampicin. You need to end your transcription process with the drugs and the disease. This is what I asked you. Where does RNA rifampicin act? RNA polymerase, prokaryotic RNA polymerase, not 30S subunits. 30S subunits are your aminoglycosides, inhibitors of 30S are your aminoglycosides and your tetracyclines. They're different. It's good to do replication versus transcription, but please note replication is not being tested in this exam. Understand that. Did we distinguish RNA polymerase 1 products from the others? 1 products are usually in the nucleolus. Is 5S formed by 1? Is 5S subunit formed by RNA polymerase 1? No, by 3. So it's not in the nucleolus. Then we discussed the post-transcriptional processing. Then we discussed the post-transcriptional processing. Please understand, everyone is not undergoing extensive post-transcriptional processing. Ribosomal RNA is only cleaved. Once it's formed, it's cleaved so that you have balanced subunits of ribosomal RNA. Not every RNA is undergoing extensive post-transcriptional processing. And post-transcriptional processing for ribosomal RNA is more splicing than modification. And did we discuss this template independent, which is the addition of CCA tail? There is some cleavage on 5 prime, 3 prime side. It's always like that. Please remember, whenever any protein, RNA is synthesized, they are usually synthesized with a more, and then they are cleaved to the final product. But yes, adding the CCA is template independent. Cleaving on 5 prime, 3 prime side is also template independent. But why this is important? Because this is the carrier of, this decides. This is the place which attaches what? This is the CCA sequence which attaches what? Why is the CCA sequence on 3 prime end of transfer RNA so important? It attaches the amino acid depending on whatever is the codon. It has to attach the same amino acid so that same amino acid can be incorporated in the peptide. Co and post means co is that modification is starting as synthesis is going on. Post means modification is starting once the synthesis is complete. Both happens in transcription. Cap on 5 prime side, tail on 3 prime. There is a signal. There's signal in life too, right? Where to go. Similarly for them too. Please understand, for splicing, don't Google too much. You have 5 prime GU, 3 prime AG. 5 prime is exon intron border. 3 prime is intron exon border. So tell me, if you have a mutation of 5 prime site, what will be the resultant transcript? Again, 5 prime GU, 3 prime AG, 5 prime exon intron, 3 prime intron exon. So if you have a mutation on 5 prime site, what would be the resultant transcript? Anyone? 5 prime site mutation, which is GU. Resultant transcript will be longer or shorter? 5 prime. Anyone? Application. Not shorter. 5 prime is exon intron border. Yes. So now more will be included. Longer. It's longer. When it's 3 prime, which is intron exon border, then the resultant transcript is shorter. Does that make sense? For that, you need to understand what comes first, exon on 5' side. Yes, Joel?
Sorry, could you explain why the five, if you cut the 5' intron, it would be longer? I kind of don't get that.
On intron. What is 3' intron exon? I ask you, what if there's a mutation on the 5' side? Then you will not recognize this border. What will happen? Are exons included? Are exons included?
S-
Are,
Sorry,
are exons
sorry,
in, are exons included or excluded?
uh, can you repeat what you just said? I just had some internet issues. I'm sorry.
I said if there's a mutation on 5' site and you don't recognize the exon intron border, are exons normally included in splicing?
No?
So are
Or,
intron included? Who spliced? Intron or exon in a normal splicing phenomena?
oh, introns.
So yes, so are exons included
Oh,
in the
then,
final transcript?
oh, in the final transcript, yes.
Yes, but since you have a mutation here and there's no recognition of exon intron border, now more will be included. So
Oh,
the resultant transcript will be longer.
okay, thank you.
Great. Let's go further. So identify both sites. Understand what is 5' GU versus 3' AG. What is exon intron versus 5' versus intron exon. Then also bring in diseases, which is Hutchinson's SLE. So this is about RNA. Let's do codon. Yes. You have any question? Anybody has question? I'm just refreshing this. So if you have not viewed anything, okay, why 3' end would be shorter? Because 3' end is AG, which is intron exon border. If there is a mutation at this site and you lose the intron exon border, normally introns are excluded during splicing. Now more will be excluded and the resultant transcript will be shorter. Amir, did I explain you that? Great. What happens in mutation in the branch point? Now that's very tricky because remember that the branch point, when you say splice sites are more specific with border. When there's a branch point, splicing is not even happening. Does that make sense? Great. So let's do genetic code. Here I'm going to only refresh you. I taught you in details. I'm not teaching you. My pointers are same as your learning objective, but see, you have a list of, I've just tried to make it more specific. You have a list of trinucleotide expansion. So do you need to ICE genetic code with trinucleotide expansion disease list? Yes. Translation read through drugs? Yes, which I mentioned here. Cystic fibrosis? Yes. Huntington's disease? Yes. So that is why this list might help you just to see. You don't need to go over this list that before the exam, did I miss anything on this list? That's all. So, and just same as your learning objectives. Know the codon properties. Know that there is more than one codon for an amino acid, which helps and is protective for mutation. And that is why you should know mutations well. So I'll just go over mutation. Otherwise, you know what is degeneracy. You know what is redundancy or degeneracy of codon. You know what swabble hypothesis. You know how to read now codon, anticodon. If they give you anticodon, you can easily find out codons. They are complementary and anti-parallel. But understand their polarity with respect to each other. Yes, application question may be there, right? Please understand there are only 15 questions. So you got to be focused here, right? Do you need to know mutations? Absolutely. Point versus versus trinucleotide versus frame shift versus non-frame shift. Please do cystic fibrosis, which is non-frame shift. When you talk about trinucleotide, that is in my next section, you should know disease under trinucleotide. Who will tell me disease under trinucleotide expansion? What diseases do you know? What are Huntington's disease? Anything else? Huntington's? Fragile X? Trinucleotide is not Alzheimer's. Please, let's be more specific. Let's not Google that list. Stick to your slides. It's fragile. It's Huntington's disease, Fragile X, uh, uh, no myotonic dystrophy. That's different from Duchenne's and Becker. The dystrophy, which is trinucleotide, is not Duchenne's and Becker's. That's known as myotonic dystrophy. So your examples are Huntington's disease, Fragile X, myotonic dystrophy, and Friedreich's ataxia. Very commonly, students are Friedreich's ataxia, very good, someone added. So these are the 4 disorders, your Huntington's disease, Fragile X, myotonic dystrophy. That's not Becker's and Duchenne's, that's different. Then the last one is Friedreich's ataxia. So examples of disease and why these mutations are different from each other. Please pay attention on them. I have done it very well with your questions. This is what I was pointing towards, trinucleotide expansion. Huntington disease is in the coding region. That's why, what does CAG code with in the genetic code table? Anyone? What does CAG code in genetic code table? What does CAG code with? Glutamine. The 3-letter word for glutamine is GLN, but single letter word for glutamine is Q. That's why it's also known as polyglutamine residues that are deposited, and also known as poly-Q disease, and is neurodegenerative. So understand, if it's in coding region, naturally you will have multiple amino acids incorporated. But non-coding region, 5' UTR, Fragile X, please don't do details. We will do details in the upcoming lectures. But 5' UTR, Fragile X, 3' UTR, myotonic dystrophy. This is not Duchenne's and Becker's, and introns, Friedreich's ataxia. Why am I bringing this? Are they non-coding region? 5' UTR, 3' UTR, and intron. Are they non-coding region? Are they non-coding region? Which one of them is not present in the mature transcript? 5' intron. So see, you got to understand what is coding, what is non-coding, what is 5' UTR, what is 3' UTR, where are they derived from, where are promoters, 5', where are enhancers and silencer, 5' or 5' as well as 3'. Enhancer silencers, 5' or 5' as well as 3'. Enhancer silencers, they are very distal. Are they on both 5', 3'? Both. They are both 5', 3', but promoter is only 5'. So that's why, did we do Huntington's disease, polyglutamine? Because it will, and a CAG repeats, CAG encodes for glutamine. No, enhancer silencers are both, not just 5' UTR. Enhancer silencers can be 5' UTR or 3' UTR. They are very distal to the promoters, but promoters are only 5' side. So we did Huntington's disease, but I also addressed other non-coding trinucleotide repeat expansion, which is your 5' UTR, Fragile X, 3' UTR, myotonic dystrophy, and intron, Friedreich's ataxia. But we did Huntington's disease. You know it is in the coding region. You know CAG codes for glutamine. Glutamine, single letter word, Q. So you have a Fragile X, myotonic dystrophy, and Friedreich's ataxia are non-coding trinucleotide repeat. Trinucleotide repeat means you have repeats happening at occurring because of mutation a number of times, but only in the coding region they result in incorporation of different amino acid. For example, CAG repeats will incorporate glutamine. Please understand the pedigree as opposed to the gel. 40 and above, we discussed it in Foundation 1-2 in Huntington's disease should express the phenotype. Our case of interest was MP, who had the 19 and 43 expansion, and the daughter was DP, who had the 16 and 43 expansion. So there was the person of interest. So this is pedigree. This is the lane corresponding to the dad, and this is the lane corresponding to the daughter. Daughter did not show was currently healthy. That means she may develop the disease later, because I think daughter is not age 40 or 35 and 40 range. We are not testing you on genetic concept, but we are definitely testing you on trinucleotide repeat expansion. It is good to now connect trinucleotide repeat expansion with the pedigree. That is why these lectures are taught in the same way. BP was the brother, which had no phenotype. We had MP, which had the phenotype. We had DP, daughter, D for daughter, B for brother. Because I don't know why we have this MP, DP, BP, but anyways, B was brother and D was daughter. The only thing is daughter had the expansion, but she did not express the phenotype. She may develop it later because she has expansion. And only remember, this is autosomal dominant, only one allele should show expansion. You don't need both the alleles to show expansion. Right? Does that make sense? And please understand also testing. What are you assessing? You're assessing the repeats. What are the repeats in Huntington's disease? CAG repeats. CAG codes for glutamine, that too in the coding region, right? Then we did all the type of mutation. Please bring them. Point is silent, no change. Now here, please understand. First, you need to know mutation, but don't get stressed with the application question. If they give you template strand, just quickly find out the mRNA strand. mRNA is complementary to template, and then read from 5' direction. No change. Please understand there's only one goal on this test, is to find out mRNA sequence. Whether they give you sense, whether they give you template, or they give you anticodon, or they give you codon. Your aim is mRNA codon and go to the genetic code table. I hope you know how to read them. Sense always 5', template always 3', mRNA always 5' to 3'. 5' corresponds to N, 3' corresponds to C. We have not gone there yet. It is in regulation of gene expression. Please do the interactive session questions and practice question so that your application questions are full grade and credit to you. Does that make sense? Because all you need to do is they give you sense strand, find out mRNA. Template, find out mRNA. Pay attention on polarity. Pay attention on what is given. If template is given, you have to find complementary for mRNA. If sense is given, you're not finding complementary. So don't make those little mistakes. If you are given anticodon for mRNA, do you have to find out complementary? If anticodon is given, do you get to find out complementary sequence for mRNA? If they have given you anticodon sequence, I'm asking you, have you zoned out? If you're given anticodon sequence, do you have to find out complementary? Yes. So understand where you have to find out complementary, where is it same, and that is where you have to decode the mRNA sequence and amino acid chain. Does that make sense? It's not very difficult. Do some few questions on the practice and you should be fine. Believe me. All don't mess that up, and the polarity, right? So when they give you template, you have to find out complementary sense for mRNA. When they give you template for mRNA, find out complementary sequence. When they give you sense, mRNA is same as sense. If they give you anticodon, again, you have to find out complementary. But remember that complementary is always with respect to others. For DNA, who are complementary? Template and sense strand. For transfer RNA and mRNA who are complementary, codon and anticodon. So you got to just read that. Yes, anyone has a question? If they give you anticodon sequence, no, not necessarily. But if they give you anticodon sequence, for example, they gave you anticodon sequence in 5' 3' direction, you still first find out the mRNA codon in 3' to 5', but read from 5'. So it is not necessary that they will always give you anticodon in 3' to 5' direction, but anticodon should be 3' to 5', right? If mRNA is 5' to 3'. Not always. Somebody asked me, will anticodon be always given in 3'? That's what you need to pay attention to, right? But one thing is sure, that if they give you anticodon, you got to find out complementary. Am I right? Did I answer you? I'm not. Did I answer you? Somebody has a question in the chat? Yes. All you need to do is understand where you have to find out complementary and where you don't have to find out complementary. Also, very important, which students make a mistake, is please, when you find complementary, polarity is already reversed. But you don't do that in sense strand. For sense strand, if you want to find out mRNA, the polarity is not reversed. If sense strand is given in 5' to 3' direction, and if mRNA is same in sense strand, that answer would be in 5' to 3' direction, right? So you need to understand respective relationship of the strand, whether it's sense template, codon anticodon, sense mRNA, template mRNA. Does that make sense? Okay, so you got to do a point mutation, nonsense, stop codons, silent, no change, missense change. Also, please go a step further in missense. Is it conservative or non-conservative? For that, I have given you amino acid chart. If they are of the same class, conservative. Just know your non-polar class, you will understand which is the rest are polar, right? So conservative means the change was from the same class. For example, valine was replaced by leucine, conservative, right? If valine replaced by aspartic acid, is it conservative? Valine replaced by aspartic acid, is it conservative? Valine replaced by, no, and valine replaced by aspartic acid. What kind of mutation is this? Point mutation? It is non-conservative, but is it point mutation, trinucleotide, frame shift, or non-frame shift? Point, trinucleotide, frame shift, or non-frame shift? Point. Is it missense, nonsense, or silent? When I said valine chain, missense. So see, you got to know that classification, right? Definition, subclassification. Yes, if I'm saying valine is replaced by aspartic acid, the class is point mutation, subclass is missense, subclass is non-conservative. Don't miss the step. You would be correct in detecting mutation. This is point mutation. Is this point mutation slide? Is this point mutation? Is it missense? And is it conservative? Is it non-conservative? So it is point, missense, and is it non-conservative? Yes, right? So identify the right mutation. That is your goal. That's all. By definition, you know point is single nucleotide change. Trinucleotide is you will see repeat expansions, know the disease. Frame shift, you will have, they are not insertion and deletion, first of all, not change, insertion and deletion, not 3 or multiple of 3. Non-frame shift, 3, insertion or deletion. Yes, anyone has a question? Yeah, non-conservative. It's not conservative. And non-conservative. It's not about asking I'm conservative or not. I'm just joking. I know you're saying non-conservative. So the other thing is also understand what is the mutation if levels are normal, size are normal, but activity is reduced. For example, if the activity was normal, size was normal, but levels were reduced, what could be the mutation? If the size was normal, the activity was normal, but the levels were reduced. What kind of mutation, if I narrowed it down, was it missense, silent, or nonsense? No, activity was okay. The no, please listen to me. I'm saying the size of the resultant protein was okay, activity was okay, but the levels were reduced. What could that be? The cause of this outcome could have been which mutation? Missense, nonsense, or silent? Yeah, if it's silent, why would the activity be reduced? No, missense activity should be reduced. Levels. Okay, go back again. I'm saying if the levels, hear me again. I'm saying if the resultant mRNA or protein is the same. When they say mRNA, that outcome is also going to come in protein, right? First, you agree with me? I'm saying if I give you another scenario that size was okay, activity was okay, but the levels were reduced, then is it missense, nonsense, or silent? But missense change, what if missense change was conservative? How can that reduce the level? Missense will affect activity. Levels will be affected by what? What can be introduced that the levels could be low? Premature stop codon? Yeah, a stop codon or a nonsense mutation. Yeah, so I'm just talking about what could be you have to choose the best answer, right? If levels are reduced, then it could be a stop codon. Yes, it could be E. No, E would be size. And now we are going everywhere. I want to finish this. Okay, let me tell you. So somebody's saying, let's look at this choices, right? I asked you another scenario that you have normal levels, you have size okay, activity okay, but the levels were reduced. Then you would choose nonsense. That's all I'm pointing you to, right? Somebody said deletion. No, deletion will not result in low levels. Deletion will result in the size will change. Insertion, it will be longer. Deletion, it will be shorter. So there's difference between size, activity, and levels. Do you see that now? So I was just giving you another. What doesn't make sense, Diego?
No, it is what I said doesn't make sense.
Okay. I thought it's still not, you are still confused. Okay, let's go further. That's why giving you that you could understand levels, size, and activity here. And I just gave you another scenario. So do you understand that deletion of nucleotide will not result in low levels, please? Deletion of nucleotide will change the size, not the levels. Do you understand these different things, which is level, size versus activity? Yes? No. Sorry, I want you to be clear because we are doing a review. You have exam. You need to learn it now, right? Level of transcription. Yes, level is coming only when you prematurely stop it. Then the levels are reduced. Yeah, nonsense would be stop codon. But now listen this carefully. Somebody has a good question. Now, point mutation, don't mix them, please. Point mutation, trinucleotide, frame shift, non-frame shift, okay? Point mutation is single nucleotide chain. Because in frame shift also there is premature termination of codon. That's not nonsense, okay? In frame shift also there is premature termination of codon as an outcome, but that is not nonsense. Nonsense mutation means that will come under point. If there's a single nucleotide chain and that change is stop codon. Now let's go to somebody has a question on premature termination of codon, which is also seen in frame shift. Yes, frame shift because there are two outcomes. Either the whole reading frame is changed or there is premature termination of codon. That is because of stop codon only, but that doesn't come in point mutation. Do you understand the difference? Yes? Do you understand the difference? Do you understand the difference in outcome of stop codon can also be present in frame shift, but that doesn't mean it's a nonsense mutation, right? Yes, because a stop codon came and there was premature termination of codon because of change in the reading frame. But that doesn't mean that is nonsense. Nonsense is still. So the mutation is dependent on the definition of the class, not the subclass or the outcome. Please understand that. Yes? Does it make sense? We can discuss the question later. Let me finish this because I think I want to finish in 10 minutes. So let's pay attention, right? So that at least you know what are the we will discuss nonsense. Give me a minute. We have the next hour open for question answer, right? I will tell you what there could be stop codon in point two and stop codon in frame shift two. I'll make you understand the difference, but let's go further, right? Frame shift is one or frame shift is three and not multiple of three. Please, if they give you sense strand, find out mutation. It's very easy. You compare the sense with the mutant sense strand, right? mRNA is always same as the sense strand. It doesn't matter whether it's normal or mutant. mRNA is still the same as sense strand. Good to see the application exercise because we are not going to ask you first order question. What is point mutation? Yes, you have to definitely understand difference between point versus trinucleotide versus frame shift versus non-frame shift with disease example, right? Does that make sense? Please, one more thing that I want to introduce. Yes, I will take your questions. Okay, I'll open it at 11. Just let me skim towards the lectures. Please, one thing that students make a mistake is they don't understand the reading frame. So this is the first reading frame. There could be second, there could be third. Please be mindful. I'm telling the whole class. If you start from the first reading frame, it is AUG. But if you start from the second reading frame, the whole codon changes. So please don't make that mistake. If you start from second reading frame, it will become UGC. If you start from the third reading frame, the codon will become GCA. The first codon will be GCA. Please be very mindful of that. Please be mindful of this reading frame 1, reading frame 2 versus 3. Your whole sequence will change and you will decode a wrong amino acid. AUG is for which one? AUG codes for? AUG codes for start codon, but codes for which amino acid? AUG codes for methionine, absolutely correct. Very good. 3 or multiple of 3, non-frame shift. Non-frame shift, cystic fibrosis. Dr. Binstock has explained this, but I'll get to this later on if anybody has confusion. That's why I got this slide. Some students have confusion between what is insertion versus deletion errors during replication. I will explain you. But non-frame shifts, cystic fibrosis, delta F508, most common deletion, no change in amino acid at either C or N terminal, please. Right? Do you have antisense oligonucleotide in this? I think I got a wrong slide. Am I right? Do you have the antisense oligonucleotide also in this lecture? Yeah, it is mentioned, right? Okay, great, great, great. So Duchenne's versus Becker's, right? So what is antisense oligonucleotide? You are masking the exon to be read. Simply, it's like that. The motive of this drug is to mask the site so it is not read. Why do you do it for Duchenne's? Because Duchenne's is severe than Becker's. You want to help Duchenne patients to become Becker. Becker is better. So you mask this by masking this. The reading frame is retained, and especially exon 51. And that helps Duchenne's patient have at least some functional protein. So masking the exon so that it's not read during splicing is done by antisense oligonucleotide, which is different from translation read through drugs. Translation read through drugs do what? They don't read what? Translation read through drugs do what? They don't read which codon? The stop codon. If you will not read them, there will be no premature termination of the chain, and you can have some functional protein. And the same aim is in the antisense oligonucleotide. You're masking the reading of this exon and changing the splice site so you can have you can retain the reading frame. That's why you help Duchenne's with patients with becoming Becker. Becker is better. Okay, so I don't know, how do students have, okay, do DOs have lab at 11? I thought our 10 to 12 time was for biochem and genetics, isn't it? Am I right or no? Okay, let me finish this. Whoever is there, please. There's a lot of miscommunication about sessions and times in the present semester. Maybe because of the new curriculum, but we got to sort this, right? Okay, great. Let's go on. Let's move on. Whoever is in the lab, good luck. Okay, so what is antisense oligonucleotide? They are used to treat Duchenne's patient. You mask this nucleotide, so this is not included and not read by splicing during splicing, and that helps in maintaining the reading frame and some functional protein in Duchenne's. That is why they convert Duchenne's. It's not curing those patients. It's just helping them have at least some functional protein and help them with their signs and symptoms. So this is antisense oligonucleotide. Again, icing on the cake, which is different from translation read through drugs, which is non-stop codon, remember, and helping retaining the functional protein. I told you your trinucleotide repeat coding region, Huntington disease, non-coding, Fragile X, myotonic dystrophy, and Friedreich's ataxia. Let's quickly do protein translation so I can help you with questions. I think is it a TA session for MSK? I don't know. What are you?
Yes, that's why people are going to lab today. There's a lot of TA sessions in the Angel Lab.
Oh, so I think there's really a lack of coordination among everyone, that what sessions are going and when they need to hold them, I think, right? I think the communication got to be more broader than just between few people. But anyways, let's move on. Let's be positive, okay? We can sort this out. Let's do, because remember that these 15 questions, if you get these grades, you are good in biochemistry and genetics. Why lose them? Yes? Let's finish protein translation in 10 minutes. And anyways, choice. Yeah, and I like that, Aaron. It's always about choice. Please make the right choices. Then let's move on. Translation. Please see, for translation, I explained you again these pointers. Don't forget the drugs, please. Do not forget the drugs, diphtheria, also drugs. These are all pointers are the same as your learning objectives. Once you have done the whole lecture, go and see, did I do my, for example, did I do release factors? That release factor are not in initiation, elongation, they are in termination, right? Just distinguishing the right process in the right pathway. Please, we have done signaling, do that well, signaling of protein to their destination. Also post-translation modification. We have discussed vitamin K deficiency there, and we have discussed vitamin C scurvy there. So pay attention on that. Please don't forget these polarities. That will only help you to get the right answer. Don't panic with these application questions. First, find out what they are asking, because in the end, you have to decode the mRNA, right? Does that make sense? They can also give you anticodon, but you know the relationship between anticodon and codons. Codons are where? On transfer RNA or mRNA? Where are codons? Codons or mRNA. So understand their relationship and accordingly decode them. It's not hard. If you do few questions on how giving sense, you can find out mRNA. Template, you can find out mRNA. Anticodon, you can find out mRNA. These are the only three things you need to do with your practice questions. So I'm just giving you focus here. Initiation, elongation, termination, again. N is always on the left, C is always on the right. What are the ingredients? You need first of all your enzymes. You need amino acid transfer RNA synthetase because you want to charge the transfer RNA. You need peptidyl transferase, which is in your large subunit. You need initiation factor, elongation factor, release factor. You need energy. Some processes are energy independent, but this also has initiation, elongation, termination. Where is amino acid attached? At the 3 CCAN 3' end. What is initiator codon, initiator transfer RNA? Where amino acid is methionine. What is amino acyl transfer RNA, which students are confused with? A transfer RNA which brings any other new amino acid. You call it amino acyl transfer RNA. But if it is with methionine, you call it initiator transfer RNA, because initiating codon is AUG, right? All are energy dependent except process and translation, except peptidyl transferase, right? You got to know these structure, don't draw them, identify them. Understand that unless you don't have a large subunit, you cannot form these sites. These sites are formed only by large subunit. Initiation is different from elongation because initiation is complete. This is not initiation complete, please. Initiation is only complete when small subunit, mRNA, transfer RNA, and large subunit come together. Then you got to do drugs. Some drugs affect 30S, which are your streptomycin, aminoglycosides, doxycycline. Some drugs affect 50S, which is chloramphenicol, macrolides, and lincomycin. Don't flip-flop them, right? Yes, anything that affects 50S will affect which activity? Scanning or peptidyl transferase. So who scans? Not the large subunit. Who scans? Small subunit. So when I'm saying PEP50S, drugs affecting 50S affects peptidyl transferase activity. Where is peptidyl transferase activity? Large subunit or small? Where is
Large.
large? Please understand that large is not scanning. Scanning is done by small. And also please understand the pro and eukaryotic differences. If it was eukaryote, what would have scanned the mRNA in the initiation? Which subunit? If it was eukaryote, which subunit would have scanned here? Are you seeing my slide?
FMET.
Sorry? Which subunit? 40S in eukaryotes and 30S in prokaryotes. So there is a difference. Here it's not, it's 15 prokaryotes. What would have been in eukaryotes? If it's 60, right? If I say 70, is it pro or eu? 70. 70 it is pro. Understand that. Pro and eukaryotic differences you got to bring in, right? But initiation is complete when transfer RNA, mRNA, small and large subunit all join. Elongation is welcoming the new amino acid transfer RNA and transfer of peptidyl chain. Transfer of peptidyl chain, peptidyl transferase is only the one process which is energy independent. You got to distinguish 30S versus 50S. And also, please understand when small subunit is not reading properly, some of the antibiotics also cause misreading, which is aminoglycoside. This will not happen with large subunit. Large subunit does not scan. Large subunit has what activity? Enzymatic activity. So misreading outcome is not by drugs affecting large subunit. Misreading outcome is by drugs affecting small subunit, and especially with aminoglycoside. So you got to pay attention on the drugs, absolutely. Translocation is moving the peptides a clock back. We have done diphtheria that inhibits translocation. Termination, how do you identify termination? Anywhere you see what? So tell me, how will you identify this is a termination? The translation is at the level of termination. Anyone? How will you identify that? Just tell me one thing that will tell you that, oh, this is the translation is at the level of termination. What is one unique thing? Stop codon at the A site. But one thing is stop codon will be so hard to read. What is one thing that is very easy to see? Release factor, very good. Somebody said that, Andrew. Great. Release factor. Release factor is that means you're not in elongation. If I say EF2, where are you at? If you see EF2, what process are you at? EF2, not EF1. Elongation is not the right answer. I'm saying EF2. Translocation. EF1, elongation, right? Does that make sense? So identify the right factors, right enzyme, right processes. That's all about translation. Some energy dependent, one which is peptidyl transferase, energy independent. Please be mindful of the signal recognition particle or peptide, which is for protein synthesized by the which is by the protein synthesized by the ribosomes on the ER, known as rough endoplasmic reticulum, right? You need a signal sequence. You need a signal sequence to move. You need a signal sequence, sorry, to move the protein from ER to Golgi to outside. You need additional signal to read to direct it to lysosome. So you must know different signaling. Then, sorry, should be mindful of types of energy. No. You don't need to know ATP, GTP. Please don't waste your time there. You don't need to know even initiation factor 2, 4. Yes, you need to know EF1, EF2. Yes, and differentiate it from eukaryote, but you don't need to know ATP, GTP. No. Please see, even collagen is synthesized with a signal sequence because I told you the sole purpose of protein to be synthesized on signal sequence is that it should be exported out. And that, how does it do that? By protein synthesized by ribosomes, and the ER moves to ER, Golgi, and outside. So understand this is also for collagen. Here only I'm addressing the post-translation, which is different from post-transcriptional processing of collagen, hydroxylation that requires vitamin C intracellular, versus this we will learn in MSK2. Copper extracellular. Usually these two are asked together. So students distinguish, very important. Intracellular versus extracellular. We have not learned copper, but you have learned vitamin C intracellular, hydroxylation, strong collagen. So understand signal recognition, particle peptide sequence, and that moves proteins into ER. And then post-translation modification, we have done all of them. Hydroxylation, we have done vitamin C with it, right? Which is vitamin C deficiency. We have done vitamin K. Vitamin K deficiency will not help, will not activate clotting factors. This carboxylation, please be mindful, does not require biotin. This requires vitamin K, and the process is in the liver. So post-translation modification are very important to make proteins active. We have done hydroxylation, carboxylation, acetylation, and phosphorylation. 4 we have already done: hydroxylation, carboxylation, acetylation, and phosphorylation. Right? Have we done? We have also done methylation though. Where have we done methylation? Anyone? Methylation, as we have done with histone. Very good, very good. So some of these modifications that you have done, otherwise you don't need to memorize the whole list with. Please, hydroxylation, you got 2. Carboxylation, yes. Acetylation, yes. And phosphorylation and methylation last. Same list. Chaperones help in folding. Misfolding leads to misfolding. Proteins have to be degraded in proteasome. I want to bring your attention to cystic fibrosis CV. This is what I was telling you, that the delta F508 deletion, was it frame shift or non-frame shifts? I think this is my last slide, if I'm not wrong. Yeah, let me finish this. So we had 2 CVs. I did Huntington's disease, the MP, BP, and DP. But the second case is cystic fibrosis. So what is cystic fibrosis mutation? Frame shift or non-frame shift? What is cystic fibrosis mutation? Frame shift or non-frame shift? Well, was it frame shift? It was non-frame shift. You had deletion of 3. Whenever you have deletion and insertion of 3 or multiple of 3, it's non-frame shift. And in this disease, there was no change in amino acid, which was the most common mutation, delta F508. Look at this. That results in a misfolding protein, actually, which is degraded. That's why cystic fibrosis happens. I want to bring your attention to now what all we have learned. Though cystic fibrosis, I'll tell you, most common mutation, delta F508, but other mutations are also seen. That results in misfolding protein, which is degraded in the ER. That's why low CFTR protein in cystic fibrosis. Signs and symptoms, sweat chloride test positive, pancreatic insufficiency, and Pseudomonas infection. These are the three cardinal buzzwords for cystic fibrosis. Pancreatic insufficiency, Pseudomonas infection, susceptibility, and sweat chloride test positive. But mutation more common, delta F508, which result in misfolding protein. This compared to other mutations. So let's see this one, and you will tell me what will help. So in the first mutation, what is happening? There is very less protein. Now, what can that be due to? What could have been introduced that there is very less protein? What could have been introduced? Yes, nonsense or stop codon. And that's why in first type of mutation is most dangerous, very less protein. Second mutation, most common, resulting in misfolding protein, again less protein. But the reason is different. So this will also tell you mutation. This mutation is there is either stop codon or there is premature termination of codon. But the protein is low. This is the mutation which is non-frame shift, where there is loss of only one amino acid, but the resultant protein is still not functional. What are the other type of mutation, which is this is 1, 2, this is 3, 4, and 5. In 3, 4 is more fault with the function, either regulation or conduction. And in the last one, there is not enough CFTR. So this is very low protein. This is frame shift, non-frame shift, misfolded protein. 3 and 4 are defect in conduction and regulation rather than the quantity of protein. And this one is not very low protein, but not enough protein. So this also, so cystic fibrosis also helps you understand mutation. That's all for today. Good luck. Anyone has question now? Yes, I will. These files are going in your MSK1 exam help folder. Good luck, everyone. We will do exam review next Friday. Great.
Dr. Bach, I just have a quick question.
Sure.
Would you mind just going over again the exon skipping, that concept for, I think,
Okay.
patients?